Hello, my name is Vaishnavi Gopal and I recently finished my BSc Biomedical Science. I'm thinking of starting my master's in January, and the project I've been working on is titled The Impacts of YWHEB on Oncogenic Phenotypes in Breast Cancer, a Potential Tumor Marker. The reason I'm interested in breast cancer is because 1 in 8 women will be diagnosed with breast cancer in their lifetime. The current way of diagnosing breast cancer is by doing mammograms, and our lab wants to find a less invasive way of detecting breast cancer, and that would be by looking at certain blood biomarkers that could be detected in blood samples of patients. And specifically, our lab focuses on these oncogenic microRNAs. They're small molecules that are found in the nucleus of cells, and they are found to play roles such as metastasis, causing cells to move from their local area to other parts of the body, angiogenesis, oxidative stress and hypoxia, so other aggressive cancer phenotypes. But what was very specific to my project is that the precursor forms of these microRNAs that are supposed to be in the nucleus were found to be significantly high in the blood of patients. So then we wanted to look at these effects, and so we studied other markers that were present in the blood that were potentially causing cancer to progress. Specifically, my focus is on YWHAB. It is a protein that has many different binding partners, and it is part of the 14-3-3 family, so it plays a role in allowing cells to divide and grow. And so we wanted to see if it's promoting metastasis and causing tumor progression by altering the tumor microenvironment. And as well as we wanted to see if it could be a potential biomarker in the future. So we did several different tests. We found that it was expressed super high in aggressive breast cancer cell lines compared to poorly aggressive breast cancer cell lines. We also found that it promoted cellular migration and caused cells to evolve from epithelial to mesenchymal transition. We found that based on by looking at different databases, that it was high in tissue samples and plasma samples of cells, so in their blood and in the tissue samples. And then we used our own samples that we have in the lab to look at whether it was high in patient samples that we have from the Ontario Institute for Cancer Research. And we found that in the control sample, compared to the tissue sample, it was significantly lower, which means that it could be a potential tumor marker. So then from there, we wanted to see whether it could help detect specific breast cancer subtypes, and we found that it isn't very specific. It is high in all the subtypes. And then we wanted to see whether it was high in specific stages, and we found that it was high in all the stages. So it's not a very specific subtype or stage marker, but it is a specific tumor marker, so it can tell you have breast cancer. And then we went on to look at whether we could help detect this marker in patient samples, in breast cancer patient blood samples, sorry. And so what we did was we looked at breast cancer patient blood samples compared to benign or healthy patient samples. And we found that in the blood samples of patients with breast cancer, it was higher, but it wasn't significantly higher. So we once again broke it down, and we found that luminal A and luminal B cancers, so these are subtypes that contain the estrogen and progesterone receptors. They're not very aggressive, but we found it to be high. And then more specifically, we found it to be high in stages 1, which we wanted to look at to help detect breast cancer at an earlier stage. So what we found with this overall project is that it is a specific tumor marker, but it is not a specific blood biomarker. However, in combination with the prime microRNA that I mentioned up here, it could be a sensitive blood biomarker. And this paper was recently published in the Breast Cancer Targets and Therapy Journal in December 2024, along with my other colleagues here that helped me. So I'd like to thank you for listening and thank them for always helping me.